Good afternoon. My name is Michael Maguire. I'm CEO and co-founder of Avectas. Avectas has developed the Solupore cell engineering platform, and I'm looking forward to introducing you, or indeed to updating you on the progress of our platform since we presented a previous meeting on the meds. I'd like to thank the organisers for inviting me to speak and look forward to sharing the progress in our work with you. So Solupore is designed to enable the next wave of cell-modified gene therapies. It's terrific that we've seen now 4 approvals in cell-modified gene therapies, and these approvals are typically in products that have a single transgene modification. We view, or it's our anticipation, that the next generation of gene-modified cell therapies will be more complex. So to address allogenicity and to address solid mass tumors, which are 2 big needs in the market, we will need more complex gene editing. We will need the capability to have viral and non-viral hybridization or interaction. And lastly, the functionality and the viability of the cells will be absolutely critical for reasons which I will share. Solupore addresses all of these challenges and has been designed from the ground up to address these challenges. A little bit about the company. Avectas is based in Dublin in Ireland. We've research sites outside of Dublin and in Toronto in Canada. We also have a chief business officer based in Cambridge, Mass. We've 34 employees today. We've an experienced board and leadership team. We've a world-leading scientific advisory board, and we've raised in excess of $40 million to date. The technology that we've developed is transformative and yet simple, and we will speak to how this will address the emerging needs for next generation cell therapy. And lastly, our business model is a partnering model where we want to engage with therapeutic developers and with top tier CDMOs to bring our technology into use, to be adopted, and ultimately to confer benefits on patients. So the Solupore device, the SUS, is pictured in the center of this slide. The SUS is a GMP-aligned closed system. It's automated, software controlled, has highly reproducible results. It delivers the range of cargoes that you would imagine of a non-viral delivery platform. We deliver messenger RNA, RNPs, CRISPR-Cas9 and other gene editing tools, and also plasmid delivery as well. The system is simple and straightforward to configure and to use, and yet that belies a facility to effect very complex gene editing, very complex process development activities on this platform. And the process development folk watching this presentation will no doubt observe the potential within this platform for cell culturing as well. This is something we've thought deeply about and we think has rich possibility within this platform. We've also developed a scaled down version of the Solupore technology called the research tool. The research tool is designed to give developers a first use of the Solupore technology, if you will, to facilitate frictionless adoption. We have technology transferred in excess of 10 of these devices to sites globally where they are producing data, which we're delighted with. We've developed excellent supports for users, including tech transfer protocols, SOPs, and the like. We're also at an advanced stage of development of our continuous flow-through system. This is to handle significantly higher throughputs of cells This is something that we look forward to updating you on in the future. In terms of how Solupore works, in the transfection chamber on the left of this slide, approximately under the figure B, there is a highly engineered cell membrane or filter membrane, excuse me. And cells are introduced into this device. They're pumped into the device using the controller. When the cells come into the device in suspension, they're taken out of suspension temporarily, where the culture medium is pushed down through that filter membrane. The cells are then sprayed with an atomized vehicle which contains both the permeabilization chemistry and the cargo of choice. Or cargoes of choice. The cell membrane is transiently permeabilized, and when the mechanism of transport over the membrane is passive diffusion. When the cell uptakes the cargo, the cells are immediately resuspended. This process takes approximately 7 minutes, and it can be used as a single process, or it can be used in semi-continuous use. So there's a lot of potential for modifying or for implementing different processes. Solupore SUS has 3 core elements. There is a delivery solution, there's a single-use assembly, and there's a multi-use controller. We will file a type 2 DMF in terms of regulatory pathway on the delivery solution. We will also file a type 5 DMF on the single-use assembly and the controller aspect. Both of those filings will happen this year. Additionally, we will support users through provision of the supporting documentation to insert into the CMC section of their IND as necessary. And once again, the process development people looking at this slide will see the potential for cell culturing and implementing quite elaborate process flows within this software controlled system. This enables us to travel, I think, the whole arc between concept and commercialization, feasibilities with the research tool, then porting that work onto the clinical grade system, having the regulatory support of Avectas, and then a commercialization model that will work for partners. As you would expect with a company of our size, we have a significant and growing IP portfolio. We've also interacted extensively through partnership with a number of key institutions and partners: CCRM, Catapult, UC Davis in California, Karolinska Institute, and UCL. We've also backed our own platform, if you like, where we've entered into a partnership with Oncotherapeutics, where they have developed a dual-targeted NK therapy in which Avectas is the delivery engine. We also have a relationship with Vicelex, where we are delivering factors to NK cell cultures to promote proliferation. I'm going to speak briefly to the scientific data around the platform. So Solupore enables complex cell engineering and efficient gene editing. So on the left-hand side of this slide, we see multiplex delivery of CAR and GFP and a model cargo. What we see is high viability and excellent delivery expression. On the right-hand side of the slide, we see sequential delivery of a day 0 TRAC or NP and a day 2 CAR, transient CD19 mRNA CAR. Once again, we see high editing efficiency and we see very high viability, which is characteristic of the system. We go on to show, in terms of CRISPR-Cas9, a range of different targets, and each of them edited extremely efficiently in T cells and also in NK cells. That's on the left-hand side of the slide. On the right-hand side of the slide, we show multiple gene editing platforms. The targets are anonymized there, but within there are several, both proprietary and widely used gene editing platforms, all the time maintaining very high viabilities. Solupore has minimal impact on gene and protein expression in T cells. So we spoke earlier to a gentler technology being required to preserve function and viability of T cells. So on the left-hand side of this slide, we see a heat map at a 6-hour and 24-hour time point showing the effect on the cells in a panel of immune genes. So we see significant up and down regulation of genes in the electroporated population of cells. We do not see that in the Solupore engineered population of cells. On the right-hand side, using cytokine release as a marker of membrane damage or stress, we looked at IL-2 and IL-8 secretion. These are both elevated with the electroporated population, and we do not see that elevation with the Solupore engineered population. So Solupore is a low-stress delivery method which gives rise to effector cells which are highly viable and highly functional. We went on to demonstrate this work both in vitro and in vivo. So on the left-hand side of this slide, we're looking at an excelligence assay. We're looking at effector T cells, which have been modified with, transiently modified with mRNA CD19 CAR. So at very modest effector to target ratios between 0.1 to 1 and 2.5 to 1, we see excellent cytolysis in vitro. So we can see that these cells which have been synthesized using Solupore are highly functional and they're viable. On the right-hand side of the slide, we infused these CD19 CAR T cells into an animal model. This was a study done at Jackson Labs for us. And at the highest dosing level, 4 million cells, you can see in the green box there, we have affected complete tumor clearance in the 3 mice in the center of the bottom right-hand panel. So there's evidence, so this evidence of disease-free mice at the highest CAR T dose demonstrates once again highly functional effector cells synthesized as a result of the Solupore process. So in summary, Solupore technology is very well tolerated by cells. It has increased efficacy and higher proliferation post-processing. For autologous therapies, it enables delivery to fragile and short supply cell types, T cells, subpopulations of T cells, for instance, TILs, and NK cells. We can also effect non-viral engineering post-transduction, lentiviral transduction, for instance, or pre-lentiviral transduction. So that hybridization is a very exciting potential within the system. We can affect multiplexed or sequential editing, suitable, for instance, for allogeneic type therapies and for addressing solid mass therapies. We could deliver a wide and growing variety of cargoes to a growing variety of cells. We are particularly interested now in moving beyond T and NK cells and delivering to iPS cells. The research device pictured on the left is available now. We would be delighted to speak with you about engaging with you and having Solupore work at your lab. And the clinical device will be available by the second quarter of this year. We have our anticipated regulatory filings by the end of 2021. So thank you very much indeed for bearing with me through the presentation. Be delighted to receive questions live or by email. Thank you very much indeed.